Drapeau. I hope I got that right. Look, he's looking more like a business magnate than a scientist. But hey, looking good. All right. Again, the whole idea about stem cells, folks, and what we mentioned in the book, in our email blast, was that everybody has stem cells. It's not one person has or the other person does not. Everybody has stem cells, and Mr. Drapeau is going to explain exactly how our bodies produce these stem cells and what you need to do. So without much further ado, Sherry, you want to go ahead and introduce him?
Sure, I would be glad to. Okay, Mr. Drapeau is an author of, as David said, the book Cracking the Stem Cell Code. He's a research scientist and neurophysiologist with over 13 years of research experience in the fields of natural foods and nutrition. While pursuing scientific research in collaboration with various universities and research centers on the health benefits of certain botanical raw materials, he has lectured on nutrition to thousands of people since 1994, presenting profound health information and insights with clarity and humor. Mr. Drapeau has been the Director of Research and Development at Cell Tech International and the Desert Lake Technologies before co-founding STEM Tech Health Sciences and becoming its Chief Science Officer. Based on research he produced and information he researched produced by numerous scientific teams, Mr. Drapeau and his collaborators proposed in 2001 a breakthrough theory on adult stem cell physiology. He is the co-inventor of a revolutionary product called STEM Enhance, and as we've said, the author of Stem Cell Theory of Renewal, and this detailed in his, all of this is detailed in his new book, Cracking the Stem Cell Code. Mr. Drapeau received a bachelor's degree in neurophysiology from McGill University, Montreal in 1987, and a master's degree from the Department of Neurology and Neurosurgery from the Montreal Neurological Institute, McGill University, Montreal in 1991. Sorry about all those neuro, things are just not rolling off my tongue. I need some more stem cells. Again, we are very glad to have him as our guest tonight, and so welcome. Are you there?
I am here. Thank you very much. Okay, well, first, thank you to you, Sherry, and to David for inviting me on this webinar. So my first experience, hopefully everything will go just fine. So like David just said, you know, the intent here is to talk about stem cells in general, essentially understanding what they are, what they're doing in the body. We have a lot of information on the media about stem cells, essentially talking about getting stem cells from different sources, growing them in a test tube, injecting them in the body, in an organ, manipulating them, doing all kinds of things to them. And here what we'll be talking about essentially is more what are they doing naturally in the body, and how is it that we can use them in order to tap into, I would say, the natural potential of health of the body. So what I would like to do is talk about stem cells, what they're doing in the body, give you enough information so that you can put in perspective everything that you may have heard in the media over the past few months or few years, and through all of this, incidentally talk about a product that I have developed, but essentially and I'll go to some chronology here of all of this, how it developed, and then it will become clear how the product is really what led us into this whole field. We didn't one day decide that stem cell was a great field and it would be great to tap into this whole concept. It's really the traditional scientific approach of observing a product and try to understand how it works that really led us into this whole field. So talking about the product is interesting just because it really gives the perspective in the context of how all of this was developed. So, so essentially, all of this started when in 1994 I started to work with an aquatic botanical called Aphanizomenon plus aqua. In 1994, that's when the Dietary Supplement Health and Education Act was passed in America, and very similar regulations were passed in other places in the world. But essentially, these regulations stated that if you sell if a company is selling any kind of dietary supplements, then the company must have scientific data to back up any claims that are being made pertaining to the dietary supplements. So I was hired by a company selling this natural product, Ephanizomenon plus aqua, in short AFA, to study it and describe the mechanism of action by which people were experiencing the health benefits. Essentially, the benefits were of three general classes: benefits on inflammatory conditions, benefits on the immune system, and benefits on mental clarity, mental energy, just an overall feeling of well-being. So we did identify very specific compounds in this plant, in AFA, supporting these various benefits. For example, on the anti-inflammatory properties, we have in this product, we have a compound called phycocyanin. Phycocyanin is the blue pigment found in virtually any blue-green algae, and it was shown to be probably one of the most potent anti-inflammatory compounds produced by the natural, by nature. We then, on the immune support, we isolated in AFA a polysaccharide that was shown, and all of this was published in the scientific literature, was shown to have very specific effects on the immune system in terms of providing immune support. And we isolated from AFA, finally, a compound called phenylethylamine, PEA. PEA is a natural compound that the brain makes naturally when somebody feels happy. In biochemistry, they call it the molecule of love. And it's a relatively rare compound in nature, or at least in a plant, where we can have access to it orally. So AFA contains a significant amount of it. And when we consume AFA orally, it does bring this feeling of mental clarity, mental energy. Deficiency in PEA have been linked to low ability to concentrate, attention deficit problems, low feeling in terms of mental energy, and it could go down to depression. You know, diagnostic tools have been developed to quantify the amount of PEA or its metabolites in the blood or in urine as a way to diagnose depression. So PEA has been linked a lot into anything that pertains to mental clarity, mental energy, mood elevation. So by discovering the presence of PEA in AFA, it clearly explained the benefits that people had been reporting on AFA. But as I was doing all this research, there were a lot of other benefits that were reported by consumers that we could not explain. The extent of these benefits were quite interesting, but probably more importantly, or more of a mystery from a scientific standpoint, was the broad variety of these benefits. People were reporting benefits on the skin, on cardiovascular function, pancreatic function, liver function, brain function, on various aspects of human health. So from a scientific standpoint, how could we explain that one single product could bring benefits touching various aspects of human health? Let me just share with you one story. I really don't want here, and I'm not making a claim that the product would be good for that specific application, but it's mostly to share the kind of information that we had that were sort of provoking us in trying to think outside of the box to try to explain what was happening and really to recognize that something was happening. It was the story of a woman when she was 12 years old. She was walking with a kerosene lamp. She tripped and she burned herself third degree in the face, chest, and arm. So she was scarred literally for her whole life. When she was about 60 years old, she started to consume AFA, and within just a matter of days, the scars start to inflame, get red and itchy, but the comfortable feeling of itchiness. So she continued to take the product, and she took a picture of herself in the mirror every week. And about a year later, she sent me the photo album, and we could clearly see the entire transition, and the last picture, all the scars were gone. So we cannot make here any claims whatsoever because this is not a study. But from a scientific standpoint, it is something that could not be denied. It was just not a testimonial from one person, but clearly in the picture here we can see that something very unusual took place for that person. So for many years it remained a mystery. We really did not know what happened there or what is it that AFA was doing in the body that could lead to this broad variety of health benefits. so it was like this for many years until a colleague of mine sent me a scientific article, the title of which was Turning Blood into Brain. And this article was describing, and this was early, I believe, early 2000, even 1999, very shortly after the discovery of a method to grow in test tube human embryonic stem cells. So what this study was reporting was, along with a number of other studies, not too many, but studies reporting in women who had leukemia, who then received radiation therapy or chemotherapy, which killed all the stem cells in their bone marrow, and then they received bone marrow transplant with stem cells coming from a male donor. And here the main difference between a male and a female is essentially the Y chromosome. It's possible to tag that Y chromosome in the body. So we ended up with female women who essentially got their bone marrow wiped out and then replaced by donor's bone marrow stem cells that came from a male. So we had stem cells with the Y chromosome, and a number of years later, when they passed away at autopsy, they essentially identified brain cells with the Y chromosome. So it was essentially telling us that stem cells from the bone marrow had the ability of going into the brain and becoming brain cells. And then soon after, the same observation was done with stem cells stem cells migrating into the heart and becoming heart cells, and soon after, the same observations with liver cells. So what we started to think about with my colleague, Dr. Jensen and I, is that if stem cells can become brain cells, heart cells, and liver cells, then first, why couldn't they become also pancreatic cells and liver cells and skin cells and retinal cells and other type of cell types in the body? It would be fairly bizarre to see stem cells from the bone marrow being able to become brain cells, heart cells, and liver cells, but have this phenomenon being simply limited to these three tissues, number one. Number two, if a phenomenon like this can be observed in the body, it would be very bizarre if it was just some sort of random phenomenon, something of that magnitude, that a cell can morph, transform itself, and become cells of another tissue. It had to be something with a purpose in the body. So that was our starting point. So we published an article in the journal Medical Hypotheses. It was published in 2002, in which we proposed very simply that stem cells from the bone marrow constitute the natural renewal system of the body. That if we have a problem anywhere in the body, and a lot of the literature came from studies on heart attack. So I'm taking this here as a model, but essentially the general model is that if there's a problem in a tissue, this tissue is going to release compounds, generally speaking, inflammatory compounds. That's what happens anytime a tissue is injured. We have compounds released by that tissue that are going to attract immune cells to kind of repair, to kind of remove infections, and that's what gives us also inflammation. So a tissue can release compounds that could very specifically go to the bone marrow, trigger the release of stem cells from the bone marrow, and then these same products would also, or compounds, would attract stem cells to that tissue, and stem cells, as they migrate in the tissue, would proliferate, differentiate, meaning become cells of that tissue, essentially leading to the regeneration or repair of the tissue. So 9 years ago, 10 years ago, this was just an idea, but it was our starting point. The starting point was these two questions. What if bone marrow stem cells constituted the natural renewal or healing system of the body? And what if AFA, its mechanism of action, was to support the release of stem cells from the bone marrow, so that by putting more stem cells in the bloodstream, more stem cells are available to migrate in the various tissues, and they will then migrate in various tissues in various individuals. So at the end, we should expect to have all kinds of benefits coming from various individuals, and that's what we had with AFA. So these two questions were really our starting point. So let's start by the first part, the whole concept about stem cells. What are they doing in the body? And so first, let's define what are stem cells. Stem cells are cells that essentially are immortal, meaning that they will proliferate endlessly in the body during the entire life of an individual. And they're primitive in the sense that they are nothing with the ability of becoming anything. The best way to understand what a stem cell is to describe any kind of cell types in the body that is not a stem cell. All the cells of the body are called, generally speaking, what we call somatic cells, cells of our tissues. There are cells that are specialized. They do one specific thing. For example, pancreatic cells, beta cells of the pancreas, will produce insulin. That's the only thing that they're going to do. Muscle cells will contract upon receiving an electrical impulse. They will contract. That's the only thing that they will do. If you have a muscle, for example, sitting on top of your liver and you have liver damage, a cell of your muscle is not going to look down, look at the liver and say, the liver needs some assistance, so let's migrate to the liver and morph into a liver cell. It's not going to happen. Every single cell type of the body, the only thing that it will do is that over time it's going to die. That's the only thing the only transformation that can happen. Stem cells, on the other end, they are primitive in the sense that they do nothing. Their role actually is into their potential of becoming any kind of cell types in the body, and they are immortal in the sense that they continue to proliferate during the entire life of an individual, so the day that we will all die, our bone marrow is still going to be filled with stem cells ready to multiply, leave the bone marrow, and go and repair various tissues in the body. There are two main sources in nature, if I could say, are the embryo called the blastula. We're talking about the 8 to 10 days old embryo. It's possible to extract cells from this area here called the inner cell mass, and then if we take these cells, it's very easy to proliferate. And then one test that was developed using embryonic stem cells is to inject them under the skin, and when we inject them under the skin, they will form a teratoma. A teratoma is essentially a lump of tissue where we can see here some teeth, what looks like a stomach or liver tissue, muscle tissue, intestinal tissue. We even see some hair here. We can have some beating heart cells, some brain cells. It's essentially a lump of tissue, entirely disorganized. It is a tumor. But that test tells us something, two things very important, is that the cell that I did inject had the ability of becoming many, many different types of cells, and had the ability to proliferate quite significantly to give us a lump of tissue. And that became essentially the test to determine if the cell that we have is indeed a stem cell. Keep that in mind because it becomes pertinent into the rest of the discussion. The other source of stem cell is adult stem cells. They're coming from the bone marrow, but they can be isolated in the blood or into many kind of tissues. We know today that the heart has its own stem cells, the liver, the pancreas, the brain, the skin. Virtually any tissue has its own stem cells. You may have here in the news that we can isolate stem cells from the skin, from fat tissue. Right now, many places in the world, they will isolate stem cells from the dental pulp. So if your kid is losing his tooth, it's possible to put the tooth in some sort of a liquid that keeps them alive. You ship that to the lab, and they will isolate the stem cells, freeze them, and keep them if you need them down in the road. As we will go on into this discussion, you will realize that it may be in itself not that valuable as an approach, but nonetheless, to understand what you get in terms of information in the media, these are the different sources of stem cells. When we say adult stem cells, it does not mean coming from an adult. Stem cells can come from the moment that we are born. A baby that is born and is, let's say, 1 minute old, the stem cells in that infant are called adult stem cells. Umbilical cord stem cells, placenta stem cells, these are all adult stem cells. If we try to proliferate them in vitro in a test tube, they do not proliferate easily, and if we inject them under the skin, they do not form a teratoma. So early on, I'm talking here 8, 9 years ago, when all of this research started, the conclusion was that embryonic stem cells have much greater capability because they proliferate easily and they form a teratoma. Adult stem cells are much lesser stem cells because they do not proliferate easily and they do not form a teratoma. But what was discovered over the years, and that is going to be essentially the bulk of this presentation, is that adult stem cells are just as capable as embryonic stem cells. The problem is that they work very well in the tissue in which they're found. So if you inject stem cells into the skin, you don't get a teratoma. What you get is a patch of skin, a patch of new skin. You put them in the heart, you get new heart cells. You put them in the liver, you get new liver cells. So essentially, they will differentiate into cells in the tissue in which they are found. So if we're talking about anything that pertains to a therapy or a therapeutic approach, and obviously adult stem cells suddenly becomes much more interesting than embryonic stem cells. So if we take all of this information here and we go and do a whole review of the scientific literature on what are stem cells doing, stem cells from the bone marrow here, what are they doing naturally in the body, and we will get something here that is similar to what is presented into this brief animation. So anytime there is an injury, a problem, something that is not functioning normally in the body. So here, this circle here is representing the blood circulation, and here's the bone marrow. So anytime that there's a problem, symbolized here by this little red dot that you see on the muscle. Again, this is just to symbolize any kind of problem that you can have in the body. This area is going to release very, very specific compounds. They have been identified. The main one is called granulocyte colony stimulating factor, GCSF. It is a compound that is very well known in medicine. For example, in used in cancer therapy or cancer treatments. If somebody goes for a full-blown chemotherapy or radiation therapy, this is going to kill all the stem cells in the bone marrow. So we need to find a donor. That's what we used to do before the discovery of all this information in the mid-80s. And what we do today is that we inject somebody with GCSF, the patient with GCSF. It triggers a massive release of their own stem cells. We can harvest these stem cells, freeze them. Then the person goes through chemotherapy, radiation therapy, and after the therapy, we give the person his or her own stem cells. So there's no problem of rejection, no problem of complication. So it is now used very commonly. So it's a compound that is very well known in science. So when you have an injury, we can talk here about a cut, a burn, a heart attack, a stroke, a bone fracture. It doesn't really matter. There's a problem or even a degenerative problem. This compound is going to be measured and going to be found in the bloodstream, released by the area of the body that has the problem. When this compound, GCSF, reaches the bone marrow, it triggers the release of stem cells from the bone marrow. So within a few days after the problem, then we start to see stem cells being released from the bone marrow and appearing in the bloodstream. They're always present in the bloodstream in a small amount, but after an injury, they will increase many-fold, anywhere, let's say, between 5 to 10-fold. And they circulate like this in the bloodstream. They don't know where to go when they're released from the bone marrow, but the area that has the problem is also going to release other kinds of compounds. The one that is very well known to do this effect, actually the only one known to have this effect, is called stromal-derived factor 1. When stromal-derived factor 1 gets in touch with stem cells circulating into the capillaries that are essentially delivering blood to this area where we have this problem, where SDF1 is being secreted. When SDF1 gets in touch with these stem cells, it triggers the expression of adhesion molecule, and these stem cells are going to leave the blood circulation, get into this tissue, and as they get in touch with cells of that tissue, they essentially are going to become cells, proliferate, and then become cells of that tissue, literally repairing that tissue. So in this whole phenomenon here, if we look back at this illustration, in this whole phenomenon, that an organ or a tissue that has a problem, that it will release compounds, there's nothing new here. We all know that any kind of tissue that has an injury will release compounds, generally known as inflammatory compounds, cytokines. The bone marrow can release cells. There's nothing new here. All the blood cells are coming from the bone marrow. That cells can leave the blood circulation and migrate in the tissue. There's nothing new here. Immune cells are very well known for their ability to migrate in the tissue where we have a problem, an infection. So what is dramatically new in this entire presentation here is the description or the observation that a cell of a type has the ability of transform itself and become cells of another type. This is dramatically new in science. So new that if you still go to med school today, what is still being taught is that stem cells, their only ability, stem cells from the bone marrow, I mean, adult stem cells, their only ability is to become blood cells. Red blood cells, white blood cells, and platelets. If anybody is talking about ability of stem cells to become brain cells and skin cells and other type of cells, this kind of information is going to be seen as very marginal and really nothing that is significant in the body. And yet what we're discovering here is that not only it is significant, but it is taking place every day of our life. It is our genuine, natural repair system of the body. So let's look a little bit. Let's look at this with just a little bit more detail. How is this process taking place? Here's a study that was done, published, I believe it was in 2003, where a group of scientists took essentially a beaker with liquid that can keep cells alive. In this beaker, they put a screen that, the property of that screen or that mesh is that the holes in the mesh are big enough to allow molecules to go through, but too small to allow the passage of cells. So it's a semi-permeable membrane. And on top of these, we put essentially stem cells. And at the bottom of which, they placed a piece of crushed liver. So that liver being injured, it is releasing compounds that essentially would tell stem cells that they need to migrate and they need to become liver cells. So they put this piece of crushed liver in the presence of these stem cells, and they observed the stem cells above. And what they saw is that within 8 hours, these cells essentially transformed into liver cells. Within 8 hours. So what is interesting in this observation here is that if we use classical science or traditional science to see this phenomenon or to study this phenomenon is going to be entirely invisible. What I mean by this is that the traditional approach is to use histology. We take a piece of this liver, we take a thin slice, we put it under a microscope, and we look at it. All that we're going to see are liver cells. And it's going to be entirely impossible to say that this liver cell that we're looking at was a stem cell the day before. If it looked anything like a lymphocyte, what we will say is that, well, there's a lymphocyte that has infiltrated the liver tissue, but we cannot tell that it's a stem cell. So this entire phenomenon has been forever entirely invisible, and it became visible with the discovery of a very interesting tool called green fluorescent protein. Like many other discoveries in science, it was possible to observe this phenomenon with the discovery of a new tool. For example, how did we discover bacteria? We discovered bacteria after the development of the microscope. It was essentially a merchant of textile that was using a microscope. We're talking here in the 1600s, 1700s, looking at a textile to be able to count thread densities, to be able to assess the quality of fabrics. He was essentially very curious, and one day he looked at a drop of water and started to see a lot of little things floating in that drop of water, and that was the first observation of bacteria. Not only did he discover that drop of water had bacteria, but he discovered later on, as we kept looking at things, that bacteria are ubiquitous. They are everywhere in the world. Same thing here. We first discovered that stem cells had the ability to migrate in different tissues and become cells of various tissues. But not only did we discover this, but we discovered that they're doing this every day in the body, in virtually every single tissue of the body, and that took place by the discovery of green fluorescent protein, a protein that is spontaneously fluorescent, extracted from a jellyfish. Since it's a protein, it is very easy to derive its DNA, and when we have the DNA, it is very easy to inject that DNA in the nucleus of a cell. So if we do that and we inject that in the nucleus of a stem cell, then what do we have? We have a stem cell who has the ability, if it ends up into a liver, for example, to multiply into a population of liver cells. The thing that is going to be special or specific to that population of liver cell is that they will all be fluorescent green. So we then take that liver tissue, put it in a microscope, use a fluorescent light, and bingo, we get a fluorescent piece of liver. So we can tell that all these liver cells actually were stem cells coming from the bone marrow that have migrated into the liver. So if we use this approach and we use stem cells with this green fluorescent protein, now they have developed red ones, we can see that these stem cells became fibroblasts, became neurons, became muscle cells, and now it has become such a great field in science that now they have made dyes of various colors. This is the retina of the Drosophila that has been stained for various type of cells, and all of this discovery or this development is so important in science that it received the 2008 Nobel Prize in Chemistry. So how does that apply in our discussion? Very simply. Here's what we did. This is a study that our team did. You take mice, you irradiate the mice, which kills all the stem cells, and then you inject stem cells that have been that contain the gene for that green fluorescent protein, and you simply we do this treatment, and then we inject a toxin in the muscle that creates an injury to the muscle, essentially destroys the muscle, and we simply let the animal recover. And what you can see here is that after 6 weeks, the entire muscle is recovered, but it's entirely green, telling us that the entire muscle has been recovered on stem cells migrating into the muscle. If the same study is being done by triggering, for example, a heart attack, we will see that the area of the heart attack or the area of the heart that is necrotic, that has the injury, is going to be filled with new cardiac cells that are green. If we trigger a bone fracture, we will see the fracture line becoming green. If we burn the skin would be green. If we trigger a stroke, the area of the brain where we have the stroke is going to become green, filled with green neurons and glial cells. So this phenomenon is taking place essentially anywhere where we have an injury. So here's, for example, a study that was done because the question here is that if we see that this is the natural process, that stem cells will naturally migrate into a tissue, then the next question is, well, if we put more stem cells in the bloodstream, are we going to then enhance that phenomenon so that it can then be used therapeutically? It could mean something in terms of regaining health or repairing some tissues that have been damaged. So here's a study that was done at the NIH by the group of Dr. Orlik. What Dr. Orlik did is that he triggered a heart attack by ligation of the coronary artery, so strictly deprived blood flow to the heart and triggered death of the heart tissue, and then let the animal recovered. And in the control group, they did not do anything, but in the experimental group, he injected cytokines, the same one that we talked about before, the famous GCSF, granulocyte colony stimulating factor, the compound that is very well known to trigger release of stem cells from the bone marrow. So we have two groups of mice. One is control, both are having a heart attack, but one is control, and the other one is a group of mice that have released stem cells from the bone marrow. So they have more stem cells circulating in their bloodstream during 8 to 10 days. When they looked at the animal 27 days later, in the control group, you can see here in blue that most of what we have here is scar tissue. No new blood vessels, 17% survival, and fairly severe signs of cardiomyopathy. In the experimental group, the only difference here is more stem cells circulating in the bloodstream. We see a complete renewal of the ventricular wall, no scar tissue, functional new blood vessels tabbing in the other ventricles, 73% survival, and quasi-normalized cardiovascular function. So we're talking here about a protocol that can reverse the consequence of a heart attack. In a world where nothing can be done for a heart attack, I believe that this is groundbreaking observation. This was published in 2001, if I remember well, so there's nothing new with this kind of information. The reason why this is not more known is that the compounds that are used to achieve this, GCSF namely, is a compound that if we use it in that fashion in humans for more than just a few days, it has very, very, very severe side effects. So other compounds are being developed, but the reason why I'm sharing this is that the concept of having more stem cells circulating in the bloodstream is extremely powerful in the sense that more stem cells circulating in the bloodstream means that more stem cells are available to migrate in different tissues. This is what is happening at one end of the spectrum when we massively promote or trigger the release of stem cells from the bone marrow. This kind of protocol here used in that study would increase the number of stem cells up to 100-fold in the bloodstream. Way superior than anything else that we can do naturally, and certainly way superior than the product that I'll be talking about in a few minutes. So the product that I'll be talking about does not do this kind of thing. So I just want to be clear here and not mislead anyone. I'm showing this kind of information to simply show the extent of what could happen if we do increase the number of stem cells circulating in the bloodstream. To kind of counterbalance that information, let's look at the study done by Tomoda. Tomoda was a Japanese cardiologist, and when people showed up at the hospital with heart attack, all he did is that he quantified the number of stem cells. AMI is acute myocardial infarction. So what he did is that he quantified the number of stem cells in people's blood and split these people in two groups. Group 1, people who had little to no stem cells in their blood. Group 2 had normal to high level of stem cells in their blood. The hypothesis here is that without stimulating anything, if all of this is true, people with more stem cells should reach greater recovery of the heart attack, or greater, let's say, tissue repair, albeit small, and that's what was seen. 6 months later, the group that had more stem cells in their bloodstream ended up having greater ejection fraction, which is a measure of the amount of blood being released by each heartbeat, which is a direct measure actually of heart function. So in all of this concept, if you want, was finally well demonstrated in a study published in 2005 where a group of scientists, a group of Warner, studied in 519 individuals at risk for cardiovascular problem. What he did is that he quantified the number of stem cells in their blood and then monitored during 1 year the occurrence of any kind of cardiovascular events. We're talking about heart attack, angina, any arrhythmia, any kind of heart problem, and then a year later correlated any kind of cardiovascular events with the number of stem cells that was naturally present in the bloodstream. So here we're talking again, no stimulation whatsoever, just the number of stem cells having in people's blood. And the conclusion was that the level of CD34 cells, these are stem cells, was one of the best predictors of cardiovascular health, better than essentially all the other predictors that are used normally to estimate what is the risk of someone's cardiovascular risks. And so, in other words, the higher the number of circulating stem cells in the blood, the greater the cardiovascular health. Of this, we can reach two kinds of conclusions. We can reach the conclusion that quantifying the number of stem cells in the blood could very well become one of the best way of predicting cardiovascular health. But to me, what is very interesting here is that if there are ways of augmenting the number of circulating stem cells, then we could increase cardiovascular health. Because today, we know what is the mechanism of action behind all of this is that, and it's the following. Anytime there's a problem in a tissue, and here the heart is probably one of the most important organs in the body with this regard, anytime there's hypoxia in a tissue, meaning lack of oxygen, that tissue is going to secrete a number of compounds, but essentially for this discussion, two are very pertinent. Number one, SDF1, stromal-derived factor 1, the only compound known to attract stem cells. Second, VEGF, vascular endothelial growth factor. When stem cells migrate in the heart under the influence of SDF1 and they get into the heart, they migrate in the heart and they get in contact with VEGF, that triggers the proliferation of stem cells and then their differentiation into capillary cells. So what we do here is that we promote the growth or the development of new capillaries, which in a tissue that is lacking oxygen is going to bring more fresh blood and is going to essentially resolve the problem and is going to eliminate any kind of cardiovascular risk or actually mitigate for these risks. So that's what is going to be the role of stem cells, the ongoing role of stem cells in the body. We need to understand that when on the day that somebody has a heart attack, that's not when the problem began. The problem began oftentimes, 10 years ago, we're climbing stairs 3 by 3. 5 years ago, 2 by 2. A year ago, 1 by 1. 6 months ago, 1 by 1, slowly. And suddenly, bingo, we get the heart problem. It didn't start on the day we got the heart problem. It started a long time ago. That's when stem cells can get into play. If we have the people who have more stem cells naturally circulating in their bloodstream can do the day-to-day repair of the tissue, which helps maintain cardiovascular health. That's sort of the conclusion of all of this, which is that bone marrow stem cells constitute the natural renewal system of the body. Simply supporting the release of stem cells from the bone marrow or having more stem cells naturally in your blood increases the ability of the body to repair itself, maintaining greater health. Now, a lot of these studies were done in mice and in animals. So a very legitimate question is this happening in real life in humans, and is it relevant? Is it pertinent to the whole story? And the answer is yes. And this can be seen in one very specific situation, and it is when men receive organs from women, or when women are receiving bone marrow transplant from men. In both situations, what we have is that we have the organ that is having does not have the Y chromosome. So the organ here coming from a woman being transplanted in a man does not have the Y chromosome, or the woman's own organs are not having the Y chromosome, but the stem cells in the bone marrow, the men's own stem cells, or those that have been transplanted in a woman's bone marrow, these do have the Y chromosome. So in either case, over time, we will have stem cells that are going to migrate in the tissue. So if we look at this story here of the man receiving a liver from a woman, because these have been studies. I mean, all of them have been studies, but the data here is quite impressive. In a study when the question was, if this is taking place, that means that if we go back into a bank of biopsies, tissues coming from biopsies done on men who received a liver transplant, and to follow up on complications and to see what's the problem, if we go back and look at these tissues, then we should see over time an increasing number of liver cells having the Y chromosome as the men's own stem cells are migrating in the liver tissue. And what was found is that within 4 to 13 months after the liver transplant, up to 16% of the cells found in the liver was the man's own liver cells. The stem cells that have come from the bone marrow and migrated into the liver. In one case of one individual, one man, who died after 4 months and a half of cirrhosis, so a chronic, very severe wound or injury to the liver, what was found is that 40% of the liver was made of the man's own liver cells coming from his bone marrow. So the conclusion here is that it's not a phenomenon that is taking place just a little bit in the body. It's a phenomenon that is taking place to a very large extent, to the point of being able to regenerate virtually almost half of one's liver within 4 to 5 months. So it's extremely impressive. So it is indeed our natural repair or renewal system in the body. Which leads us then to the work that we have done with this product called Stem Enhance. Our starting point, like I was saying before, is that we were having this product in which we people were reporting a broad variety of benefits. So our idea 8-9 years ago was that, well, in order to have such a wide impact and have touch so many different systems in the body or parts of the body, then it was possible, or at least hypothetical, that probably the way that AFA was working was by supporting stem cell release from the bone marrow, and then stem cells are going to migrate in various parts of the body. So if they migrate in someone's liver, then that person would have improvements in liver function. If they migrate in someone else's pancreas, then that person is going to report improvements into, let's say, glycemia. And in the same fashion, when they migrate in someone's retina, skin, muscles, joints, virtually any part of the body. And that's what we had with AFA. So the work that I'm going to talk about was published 2 years ago in a journal called Cardiovascular Revascularization Medicine. So I will not go in detail because it is the paper really shares and presents all the technical information. It would get a little bit complicated in a webinar like this. But essentially, in summary, here's the data that we have. Our hypothesis was that if we give people AFA, we should quantify an increase in the number of stem cells circulating in the blood if this plant does support the release of stem cells from the bone marrow. So we started to feed individuals. We did not see anything consistent until we started to use relatively large amount, 5 grams and more, and very consistently we saw an increase in the number of circulating stem cells of about 25%. So we sort of bingo discovered that that's what the product does. It does trigger a release of stem cells from the bone marrow. But in science, we need to have a mechanism of action to be able to talk about it. Otherwise, we need to think about where the scientific field was 8-9 years ago. The whole concept of stem cell was so new that at that time, simply to say that stem cells constitute the natural renewal system of the body was already quite a stretch. It was very difficult for any scientist not to just laugh at us when we were sharing that kind of information. Today, it's very well accepted. So to have a phenomenon like this without a mechanism of action is something that we could not really talk about scientifically. Plus, 5 grams is a lot of product. It would be like telling people, consume 10 to 20 capsules a few times a day. 60 capsules a day is not something that, the average person will do. So we needed to have both a mechanism of action and a way to identify the active compound and then concentrate it. And that was essentially the impetus behind the work that we have done. So here's a little bit the information that we have on the physiology of the bone marrow. or stem cells in the bone marrow. It's going to be a very simple overview, but it's to give a little bit of an idea of the work that we have done. In the bone marrow, stem cells have on their membrane a protein called L-selectin. When L-selectin is activated, it leads to the expression of a specific receptor called CXCR4. The CXCR4 receptor is very specific for a molecule called SDF1. If you remember well, we talked about SDF1 before. It is the molecule that is released by an injured tissue or a tissue in problem to attract stem cells to that tissue. The phenomenon taking place in an injury is exactly the same phenomenon taking place in the bone marrow all the time under normal conditions. So when SDF1 binds to this specific receptor, it leads to the expression of adhesion molecule that will lead to the adherence or the clinging of the stem cells to the bone marrow environment. These adhesion molecule will cut, if you want, from the membrane. So this kind of reaction here, expression of receptors binding with SDF1 and expression of adhesion molecule, is a constant phenomenon that keeps the stem cell clinging to the bone marrow environment. If we interfere with any of these steps here, we will trigger the release of stem cells from the bone marrow. Given the very mild release that we were getting with the product, the time frame of the release that we were getting, our hypothesis is that what we had in AFA was a blocker of L-selectin. By blocking this connection, we downregulate the expression of the receptor, so we get a rapid but very mild release of stem cells from the bone marrow. So under this hypothesis, we went and we searched an AFA, and that's what we discovered. We discovered that AFA contains an L-selectin blocker, and when we use a concentrate of that compound, only 1 gram of it triggers a 25 to 30% increase in the number of circulating stem cells, which is the equivalent of about 3 to 4 million new stem cells circulating in the bloodstream. So as we were trying to identify what is the active compound in AFA, we used the water-soluble fraction, the lipid fraction, the polysaccharide fraction, and then we sub-fractions all of these. But as we were doing all of this, when we tested the polysaccharide fraction, we saw an effect that was very bizarre and very, very consistent. We saw a reduction in the number of stem cells circulating in the blood. So we worked with this quite significantly, because it was totally unexpected, and for quite some time we really did not know what was happening there. But over time, what we learned is that this was actually the support in the migration ability or migratory ability of stem cells to the tissue. So the polysaccharide supports the migration of stem cells going to tissues in need. So the same plant here is providing us with 2 compounds, one that supports the release of stem cells from the bone marrow, and the other one supports their migration out of the blood into the tissue. So essentially what Stem Enhance is a blend of these 2 fractions, one that supports the release of stem cells from the bone marrow, the other one their migration out of the blood into the tissue. So we are supporting with 1 gram of Stem Enhance 2 of the most important or crucial aspects of this whole phenomenon, which is the release of stem cells from the bone marrow and their migration into tissues. So with this product right now, the next question was, well, this is interesting. We know that if we have more stem cells in the bloodstream, these stem cells are going to go into the tissues. And the stem cells released by the product are likely to go and contribute to the repair of tissues. But there's a point where we need to do a study asking the question, do the stem cells released by Stem Enhance, do they have the ability of migrating into tissues, and actually these very stem cells become cells of these tissues? So what we did is a very, very traditional, or I would say conventional, protocol in the world of stem cell research, which is irradiation of mice, which kills all the stem cells, injection of fluorescent stem cells, which leads to the repopulation of the bone marrow with fluorescent stem cells. Then we triggered an injury to the tibialis muscle by injection of cardiotoxin. So essentially the whole muscle is dead, goes away. And then we split the animals in 2. One group receives placebo, and the other group receives Stem Enhance. So we increase in the experimental group the release of stem cells from the bone marrow. And here's what we got. Very, very interesting data. In the control group, we receive no stimulation, we can see a very significant repair of the muscle, reconstruction of the muscle. But when we put more stem cells in the bloodstream, we can see a much more significant repair. So this is telling us two very important things. It is a natural process. It is happening naturally without doing anything. But if we do support the release of stem cells from the bone marrow, the repair process is enhanced. And the other part that is very interesting is that if we look in the other leg that did not have an injury, we can see that there's very, very little migration of stem cells in that tissue. We do see a little bit, but it would probably be fair to say that when one leg is not functioning properly, we do get a transfer in terms of body weight and center of gravity. So we're going to apply stress to that muscle. So that muscle is going to be slightly inflamed and will signal for inflammation and migration of stem cells. So it would be expected to see a little bit of it. But it does then demonstrate really the three pillars of this whole concept, which is stem cells are part of this natural repair process. It will happen to a greater extent if we put more stem cells in the bloodstream, and it is a directed process. Stem cells are going to go where I have a problem, not where I do not have a problem, which obviously is sort of a you would kind of say, duh, you know, it's kind of common sense. If it is a repair system in the body, if it is the natural repair system and renewal system of the body, then you don't want to repair something that is not broken. So there has to be a phenomenon to attract stem cells where they're needed and not where they're not needed. So the conclusion to all of this, I'm using this quote by Dr. Prentice at Indiana State University. Actually, today I think he's at Georgetown University in Washington. But essentially, if we focus on this bottom part here, direct stimulation of endogenous, meaning your own, stem cells within a tissue or the bone marrow may be the easiest, safest, and most efficient way to stimulate tissue regeneration. I think that where all of this is going is that in a world where right now we are isolating stem cells from various sources, from embryos, which obviously it's a whole concept and the whole controversies of embryonic stem cells. But aside from that, when we're talking about adult stem cells, isolation of stem cells from umbilical cord, from placenta, from your own blood, from skins, from dental pulp. Then we expand them in a test tube. Then we freeze them and preserve them to re-inject them later on, which is a relatively safe approach, but nonetheless exposes to possible complication. Here we're talking about a process where we don't have to do any of this. We're simply tapping in everybody's own stem cells, and more than that, in everyone's own natural, innate renewal system. So I do think that it is truly the future of stem cell research. So let's tap a little bit now into a new approach tapping into stem cells. I'm kind of opening here a parenthesis because what I want to talk about here is not, I really do not want to make the claim or make any kind of inference that the product that we're talking about here is going to do any of that. The reason why I'm going to share the few slides that are coming ahead is simply to give a clear understanding here that what I'm talking about is just not an idea on the fringe, an idea that is sort of pulled out of a few papers here right and left to kind of make a nice story. It is a very, very solid concept, the book that David talked about before, and I'll come back to it in a few moments. But that book that was just published a few weeks ago, Cracking the Stem Cell Code, has probably 400 to 500 scientific references in the book. So it's a very, very solid concept. So I want to share a little bit what other groups are doing currently in the scientific literature with this model here of strictly stem cell mobilization as a therapeutic approach. We're not talking about injection of any stem cells, simply triggering the release of your own stem cells, what kind of impact it could have from a health standpoint. So we can use different models, but the data here for diabetes is very interesting. So what the scientific group here did is that they use a male mice, so we're talking about the ability of following the Y chromosome, and they genetically manipulated the animals by inserting in the insulin gene. They inserted for the gene for green fluorescent protein. So anytime this animal starts to transcribe the gene for insulin, so a cell, for example, starts to secrete insulin, that cell will also express green fluorescent protein. So under an ultraviolet light, that cell is going to start to glow green. And they took stem cells from these animals and injected them in female irradiated female mice. So what we end up is that we end up with mice, female mice, whose bone marrow are made of male cells, so we can follow the Y chromosome, and these cells, if they do migrate in the pancreas and become functional pancreatic cells producing insulin, these cells are going to glow green because the gene is tagged with the gene for green fluorescent protein. So here's what we got. Focus here on this pancreatic cells extracted from the pancreas. If we look at these cells and suddenly we turn on the ultraviolet light, we see that this cell is starting to glow. So it has transcribed the gene for insulin. As it produced insulin, however, so what we did now, they use an antibody that can go and tag directly, specifically insulin, and we tag this antibody with a red dye, and we can see that that very cell is producing insulin. We can see that this other cell here is also producing insulin. Now we tagged it for the Y chromosome, and we can see that that cell contains the Y chromosome. So that study is telling us that naturally, without doing anything, stem cells from the bone marrow, because it's one of the stem cells that was injected with the Y chromosome, has the ability to migrate in the pancreas, transcribe the insulin gene, and actually produce insulin. The next question is, if I support the release of stem cells from the bone marrow, then could this phenomenon here have any kind of therapeutic value? And this was done in this study here, where what scientists did is that they isolated islets of Langerhans from the pancreas, which are the units in the pancreas producing insulin. So this is a normal one. If you inject in animals a toxin called streptozotocin, it is a bacterial toxin very well known to very specifically go and destroy the cells that you see here in green, the cells producing insulin. So this animal is essentially diabetic, clinically diabetic, cannot produce insulin. And they took these animals and then they injected a compound known to trigger stem cell release. So they had more stem cells in circulation in their blood for a number of days. And we can see the partial reconstitution of the islets of Langerhans, which was significant enough to reverse the clinical condition of diabetes in these animals. And what we see here was actually confirmed in humans when humans were injected with GCSF to trigger stem cell release. That was done on 19 individuals, and not every single one of them, 14 individuals, and 13 of them were for between 1 to 13 months were insulin-free. So again, these are all preliminary trials, but it's just to show that there is in the scientific literature information documenting that there is a link between releasing your own stem cells and then addressing certain clinical outcome. Again, just to be clear, these compounds are releasing stem cells with a magnitude that is beyond what StemEnhance is doing, what I described before. So all of this here has never been documented so far with StemEnhance. It's just a dietary supplement that on a day-to-day fashion is going to support the release of stem cells from the bone marrow. So what is all of this saying? The conclusion of all of this. And that's what you can find summarized in the book, The Stem Cell Theory of Renewal, published about a year ago, and this new book that David talked about, Cracking the Stem Cell Code, published just a few weeks ago. So the thesis of this book summarized in this last paragraph here, a new paradigm in wellness. By the way, as I mentioned before, there's about 400, a little bit more than 400 references in the book, so it's a very, very solid book. But essentially, what this book is telling us, the theme of the book, or the conclusion, is that every single tissue and organ of the body is constantly in a state of turnover, meaning that every single tissue of the body is losing cells and needs to replace them. For example, we have every 5 days or so, we have an entirely new intestinal mucosa. We have a new pancreas every 3 months to 4 years, depending on the data. We have a new lung every 4 years or so, new heart every 15 years or so. We can even have a new brain every 15 to 30 years. The number of years are really not pertinent or relevant. What is really relevant in this whole conversation is the very fact that there is such a thing as cellular loss in the body. And here, if we're talking about the lung, for example, whether it is 4 years, 5 years, or 6 years, it really does not matter. But there's a certain amount of time after which, if we do not have cellular renewal or tissue renewal, we would simply run out and not have any tissue and would die out of, having no lung, having no heart, having no pancreas. So we need to have tissue renewal. So what we're discovering all of this is that really health is a balance between cellular loss and tissue renewal. And loss of health is essentially an imbalance between the two, an imbalance between cellular loss and tissue renewal. You lose cells at a certain rate, and the rate at which we lose cells is really determined by the sum of a number of factors. Your DNA or your genetic makeup, past injuries, lifestyle, exposure to environmental toxins, diet, stress level in your life, or your ability to manage stress. Put all of these together, and I will say that this is overall the pressure that is applied on your body. Somebody who lives on constant stress, we can see that these people will age more rapidly. We can look at any president, for example, in the U.S., and just in a number of years, we can see that they visibly have gained like 10 years level of stress. We can all see that in our lives. It's an increased rate of cellular loss. Every single disease that exists is a loss of cells. If we lose more than 60% of the cells making pancreas and the liver, it's called diabetes. If you lose more than about 80% of the cells making dopamine in the substantia nigra in the brain, it leads to Parkinson. If you lose cells in the thyroid gland, it's called hypothyroidism. If you lose cells in the heart, you get cardiovascular problem. If you lose cells, whether over time or by an accident like a spinal cord injury, but essentially every kind of problem is due to cellular loss that is going to happen according to various factors. What is going to repair this is essentially, or balance for it, is tissue renewal coming from stem cells. So health is literally the balance between cellular loss and tissue renewal. So if we discover a way by which we can enhance tissue renewal by tapping into the potential of what stem cells can do, then it's literally opening a window or opening an avenue on an entirely new way of dealing with health and wellness. So the main question in all of this, and even here we can simply remove the word stem and ends, it's not pertinent, but essentially who can benefit from tapping into stem cells? And when we understand all of this, obviously the answer is anyone and everyone. When we understand, like I was talking about before, that any kind of health condition is not the result of an overnight cellular loss or overnight loss of function in an organ, but it's virtually always a slow cellular loss that is more important than tissue renewal, and it reaches a point where it goes beyond a certain point where suddenly the tissue is not functioning normally enough and it translates into a clinical condition. When we understand this whole concept, then obviously supporting the natural repair system of the body or the natural renewal system of the body becomes one of the greatest things that we can do to support and maintain optimal health. Many things can be done aside from the product that we're talking here in terms of stem and ends. We know that exercise is going to support the release of stem cells from the bone marrow and also from muscles. But what has been shown is that you need to do very significant exercise. This was shown, for example, with running a marathon. It was not observed if we do exercise that is less than running a marathon. So obviously running a marathon every day to support stem cell release is not a feasible thing, but nonetheless it's pretty clear that, not demonstrated, but pretty clear, or at least logical, that any kind of exercise or workout would to some extent release some stem cells. We know that stress will reduce the ability of stem cells to proliferate in the brain and other places in the body. We know that cigarette smoking is going to also reduce the ability of stem cells to migrate in tissue and proliferate. On the other end, we know that good rest is going to support the ability of stem cells to proliferate in the body. So there's a lot of things that we can do to support the role of stem cells, but in terms of actually supporting their release from the bone marrow, at this point in time, stem and ends has been the only product that has been shown to do that, natural compound shown to support this whole phenomenon in the body. So before we open it to questions, two questions here that are very, very common. So I'll just address them up front. Can stem and ends deplete the bone marrow? If we do that every day, then is there, are we going to reach that point where we deplete the bone marrow? And there are many ways of answering that question. Probably the easy answer is to link to what I was just talking about before. If this phenomenon is taking place when we simply do exercise or get good rest and a lot of things that we can do that is good in our life, then obviously we're not going to run out of stem cells. But from a physiological standpoint, the best way to answer the question is to understand that everywhere in nature, stem cell divide using what we call symmetrical cellular division. The DNA duplicates, one strand of the original DNA goes in each daughter cells, and one copy goes in each daughter cells. These two cells are identical daughter cells. This is what is taking place everywhere in nature, except in the bone marrow. In the bone marrow, what I have is asymmetrical cellular division, where the original DNA goes into one cell, and that cell essentially stays in the bone marrow. The copies go in the other cell. That cell is destined to migrate and go in the blood. So the purpose of this, it's also called the immortal strand hypothesis, and the purpose is to maintain in the bone marrow as the blueprint of any future cellular division in order to provide fresh cells for your body for your entire life. You don't want to make copies of copies of copies because this increases the risk of cancer or any kind of problem dramatically. So you want to keep your original DNA in the bone marrow, so that's why this process is taking place. So a corollary of this is that anytime you release a stem cells, a daughter cells was left in the bone marrow. So when we release 3 million stem cells, you're not suddenly lacking 3 million stem cells in the bone marrow, but you essentially maintain a very, very good balance in the bone marrow. So there are many other ways of answering that question, but essentially the answer is no. I would compare this essentially as taking somebody coming and saying, if I take a glass of water out of the ocean every day, am I going to empty the ocean? And the answer is obviously no. The potential of what the bone marrow can do here is far beyond what is being released by any kind of health ways or healthy ways of supporting stem cell release. The other big question is, can releasing or support the release of stem cell from the bone marrow support tumor growth? And the confusion here is coming essentially because what is a stem cell? A stem cell is a cell that has the ability of multiplying endlessly and of becoming various cell types of the body. And a tumor cell is essentially the same thing. A tumor cell is a cell that will multiply endlessly and has the ability of becoming any kind of cell types in the body. So they are the same thing. So it creates this sort of confusion. What we need to understand is that a stem cell that is a healthy cell, when it migrates in the tissue, will become cells of that tissue according to its own DNA, which is healthy DNA. So there's not going to be here the risk of producing any tumors. The best way to understand that is to understand that in your body, every single cell of our body is having the exact same DNA that was in the cells that we were at the time of conception, which was an embryonic stem cells. So every single cells of our body contains the potential of being an embryonic stem cell. A number of genes are essentially turned off or shut down that prevents the cells of our body of being embryonic stem cells, except for stem cells from the bone marrow that have retained this ability of multiplying like this endlessly. So really what a tumor is the cell that has in some way hijacked the DNA and turned back a normal cell into an embryonic-like cancer cells or rogue cell. So a stem cell is very different from what a tumor cell is. But in any case, without going into more of these details, the best way to answer the question is very strictly to do a study. So what we did is that we inoculated mice with breast cancer, human breast cancer cells that were green, with green fluorescent proteins so that we can follow them very easily. And then we split the group into one group was control, and the other group we gave them 10 times the amount of stem enhance that is normally recommended for people, so that if it really support tumor growth, then we would see it. Now, before we go to the results, let's talk about the 3 things here that could happen. Number 1, we could indeed support tumor growth. Number 2, nothing. The product has absolutely no effect, and we see no effect on tumor growth. But there's a third thing that could happen here, and it's a really new cutting-edge field of research in cancer research. And it's the fact that when a stem cell migrates in a tissue, we oftentimes think of these stem cells as an isolated cell, a unit cell that has migrated in tissue. But truly, stem cells function as a population. If you take, for example, a population of cells in a beaker, and you trigger their proliferation, and then you take a sample of the liquid in which they're proliferating, and then you inject that liquid with another beaker of cells that are quiescent, that are resting, it will trigger their population. Cells are talking to each other, and then you go back into these stem cells, and now you provoke them to become liver cells. You take samples of that liquid and expose another type of stem cells to this liquid, and it's going to trigger the transformation into liver cells. Cells talk to each other using chemicals. So when a stem cell gets into a tissue that may have a tumor, as it migrates in that tissue, starts to proliferate and start to become cells of that tissue, it essentially talks to all the proliferating cells of that tissue that it's time now to stop proliferating and become cells of that tissue. What is a tumor cell? It's a cell that proliferates without differentiating, without becoming cells of that tissue. If it were to transform into cells of that tissue, it would stop being a tumor. It would just be a blob of that tissue. So the third thing that could happen here is that stem cells could very well migrate in a tissue and reduce the tumor growth by pushing tumor cells off or out of their proliferative mode to become essentially just skin cells or tissue cells. I say skin cells because I'm thinking of melanoma or any kind of skin tumor, but here they would just become cells of that tissue. And here's what we got with Stem Enhance. After 6 weeks, what we could see is that we had a 30% reduction in tumor growth in the animals. Out of this, it's very important for me to say that the protocol was not designed to document anti-cancer property. It was just made to document that Stem Enhance does or does not support tumor growth. So the conclusion here is that it definitely does not promote tumor growth, and if anything, could prevent it. But again, I want to be clear here, we cannot draw these conclusions with that study, so no claims here is going to be made on the effect of Stem Enhance on tumor growth. So essentially, that's the information that I wanted to share with you and your people, David, so we can, from here, open it to question. If you're still there, and I haven't lost you, David.
I'm here. I don't know what happened to David. It's showing that he's still logged in. David, are you there?
I'm here. Can you hear me?
Yeah, now we can hear you.
Okay. Sorry, I had to press a few buttons. Oh, my goodness. This has been a lot of fascinating information, Christian. A little fast. You were on a roll. So I think, folks, you will actually have to either get the recording or something, because there was so much information that was poured out in this webinar. And again, Christian, this is fantastic. Thank you so much.
I apologize if it was too much. I hope it was not too much. Was it?
Okay. A lot of good stuff. That's what I was going to say. A lot of good stuff. So, again, the recordings are available afterwards. Anyway, this is a topic that you just I really don't think you get in one sitting. So, folks, be sure to take advantage of this after the meeting. Again, these webinars the recordings are free for members, of course. And I'm just going to say a couple more things. I want to show my screen real quick, Christian, take it back from you before we get into the main presentation. Trying to get around here. Okay. I've already just mentioned that. Again, folks, if you just joined us, his book, The Cracking the Stem Cell Code, which he spoke about. Again, this book was top 3 in 3 different categories in Amazon.com. The best deal can be gotten from there. I think they're running a special now, so you can get a good deal on the book. Now, Christian, you did mention something about a handout, a pamphlet that people could get. You want to talk about that a little bit?
Well, I don't have per se a handout, but there is the booklet that I talked about before called the Stem Cell Theory of Renewal. It is a much cheaper book. You can also get it on Amazon, and it really takes back all the concepts that I talked about today in a much simplified version. The Cracking the Stem Cell Code is everything that I've talked about today, but literally with all the science. I really tried to do it as simple as possible. The comments that I'm getting from people is that although it is technical, it is nonetheless a relatively easy read. But the other one, the booklet, really is made to be really simplified talks about what stem cells are, what they do in the body, what is this whole natural process, what is this AFA extract, and how it taps into stem cells. So it's essentially everything that we've been talking about today.
So it's more for lay people who don't want to read too much.
It would be for lay people who do not want to read too much, but at the same time, I really want to emphasize cracking the stem cell code. My main focus here was to make this information as available or as accessible as much as possible by the average reader. So it is technical, but I had my dad read it, who was a carpet seller. I had my mom read it. She has absolutely no background. I had a lot of friends read it. And although it is technical and written for scientists to really be able to get all the information, everybody who's read it so far came back and told me that, no, it was fine. They enjoyed it. So it is technical, but I would like to not scare anyone. It is not too technical.
Okay. Okay, there you have it, folks. Thank you. All right. Another quick announcement. If you want to try the STEM Enhance, again, we are not, the purpose of these webinars is to provide information. But then again, if you have a product that is, a technology that is definitely improving itself far and wide, then many people want to give it a try. So Heather has graciously agreed to give everyone her discount, and you can contact her on this information right there. If you still want it, we can send it to you anytime. All right. I have some questions of my own, Kristen, and I'm going to just kind of get back, leave this up here. Well, just to remind you folks, if you have any questions, just go ahead and type them in the box, and then Sherry and I will read them out. Okay, where does growth hormone come in all this? I mean, a lot of people claim, scientists, growth hormone experts claim that, well, many of these results can be gotten by increasing growth hormone production in the body. What do you have to say about that?
The link between growth hormone and stem cells, very interestingly enough, has been studied very, very little. There is one article published about a year ago, very briefly, talking about the ability of human growth hormone to support the proliferation of stem cells in the bone marrow, which would just make sense, you know, that it would help the proliferation of stem cells in the bone marrow. But at this point in time, not a whole lot of literature on it, but it would be, generally speaking, I would say that it would be logical to assume that it supports the proliferation of stem cells also in tissues, simply because so far when compounds like fibroblast growth factor, for example, for the skin, epithelial growth factor, hepatocyte growth factor, insulin growth factor. I mean, put any word growth factor in the body, and almost any of these growth factors have been shown to support the proliferation of stem cells. So human growth factor would be, I would be very surprised if it did not support the proliferation of stem cells, but unfortunately very little information available on it.
Got it. And so the way the growth hormone works is primarily through the stimulation of stem cells.
I cannot say that. All I can say is that it would be one way by which human growth factor is working in the body, but I cannot here say that it is the mechanism of action, it is finally how it is working. But it's probably one of the ways by which it's working, and probably a significant way by which it's working. Let me just add this, David. I think that as we continue to study the whole field of stem cells in real life, if I could say, I think that we're up to many, many very interesting discoveries. For example, I was just talking about sleep before. We know very well that sleep is very important for health. You know, the one thing that a doctor will tell you after any kind of, I don't know, surgery, treatments, therapy, whatever it is get good rest. So it's a very common thing. But what is it? What is it that you get when you rest? I mean, the role of sleep in health is not very well understood. I'm not saying that it's all that, but one thing that we're discovering right now is that when you sleep, you produce melatonin. When you have melatonin circulating in your brain and in your blood, it supports the proliferation of stem cells and their differentiation into tissue cells. We all know that stress is not good for us. If you live under stress for long periods of time, you expose yourself to the development of a number of diseases, and you just don't do good over time when stressed. What was shown is that stress, literally, we're talking here about adrenal gland hormones like cortisol, adrenaline. They do suppress the ability of stem cells to migrate in tissues and to proliferate. Cigarette smoking, the same thing. So I would say, and this is just prediction here, but I would not be surprised that as we continue, I mean the scientific community, as we continue to study what stem cells are doing in the body, I believe that what we're going to discover is that an awful lot of things that we do that are good for health, like antioxidants, for example, natural anti-inflammatory compound, already there we can explain the mechanism of action, but a lot of these things will have one of their effect in the body will be through their support of the overall role of stem cells in the body.
That's interesting. Now, a lot of the part of the lecture, the webinar, you mentioned the effects of this stem cell stimulation primarily in the bone marrow. Now, we know that there are stem cells that are resident in certain parts of the brain, like the hippocampus and the ventricular system. Can those areas that produce stem cells in those parts of the brain be stimulated, or does it have to come from the bone marrow and migrate to the brain?
No, stem cells, local stem cells will do the job, I was going to say just as well. It's not just as well, but they will proliferate and become cells of that tissue. It's just that what is most likely going to happen is that stem cells, as they migrate from the bone marrow, as they penetrate the tissue, they will first become tissue stem cells, which will then become tissue cells. So they essentially replenish various tissues in the body. So if you get some sort of a very low-level injury or just a small repair, it can be handled by local stem cells. But if you get anything that is above normal, then they come from stem cells from the bone marrow, which will then replenish the tissue cell locally, which will then become cells of that tissue. So the stem cells in each tissue are very much doing that same work. From the bone marrow is just the replenishment of these tissues. On the brain, and elsewhere also, but more specifically in the brain, or more importantly in the brain, the other phenomenon that is taking place, simply because the question focused on the brain, is that on the brain, the ability of stem cells to migrate in the brain is actually reduced, or less than in other tissues, but they secrete growth factors which are going to then activate local stem cells to do the tissue repair. So stem cells from the bone marrow are still contributing to this whole repair process, but specifically in the nervous system, a lot of the actual repair will actually take place from stem cells from the tissue.
Got it. And is the reduced ability due to the blood-brain barrier?
Most likely.
Okay.
Although stem cells do migrate in the tissues into a significant amount nonetheless, it's just that they do not seem to proliferate as much in the brain as they do in other tissues. If you look at all the new cells in the brain that came, let's say you use the same system that we used before, fluorescent stem cells. You see these fluorescent cells in the brain, but when you look at all the new cells, among the new cells, very few of them are fluorescent. So a lot of these new cells actually came from local tissue or brain stem cells, but they do migrate in the tissue nonetheless.
Got it. Got it. Is STEM enhanced safe for kids?
Yes, it is.
Okay, good.
We don't promote it for kids just because, you know, if you promote something for kids, then you need to have studies specifically done with kids, and we have not done that. But we have a lot of stories of kids taking the product, and there's really no problem.
Got it. Now, there are some couple of questions with regard to specific disease, folks. We're going to have to avoid some of those for obvious reasons. We can, though, speak in general terms, can't we, Christian? I mean, there's questions
correct.
about brain diseases, neurodegenerative disorders like you know of. So do you want to just cover that with neurodegenerative disorders?
Yeah, correct. I mean, it's obviously after a presentation like this, it's a very, very common question. You know, can stem cells or stemminants be good for such and such problem? And here, you know, I've quite literally got the whole list throughout the years. But obviously, like you said, David, we cannot answer these questions. It's a dietary supplement. It's a natural product, and through the FDA or the Dietary Supplement Health and Education Act, we cannot make any claims pertaining to the effect of a natural compound in treating diseases. Plus, you know, quite honestly, we have not done the clinical trials either to say that the product will be effective, simply because we just don't have the funds. Where you know, it's not a pharmaceutical company to do that kind of things. But from a general standpoint, what we can say, which is easy to understand, is that we know that stem cells from the bone marrow can migrate naturally into various tissues, and they can become brain cells, skin cells, joint cells, heart cells, liver cells, pancreatic cells, retinal cells, essentially any kind of cell types in the body. We know that if you put more stem cells in your blood, that means that more stem cells are available to migrate in any tissue that may be signaling, and by signaling I mean maybe sending this SOS, attracting stem cells to that tissue. So anything that we do that put more stem cells in the blood has the potential here of helping organs and tissue just functioning better. So this being said, the very objective answer is just, give it a try. There's potential, but we cannot really make any claim here. And let me just add, David, here, we did a very interesting study about a year ago. I'm just actually submitting the paper this week, the scientific paper. But people taking the product starting to report, and not one, two people, a lot of people started to report that their hair color, people were starting to get more and more gray hair. Their hair color was returning to their natural hair color. And at first I was sort of irritated by these kinds of comments. I was telling people, don't say stupid things. We don't need to state anything that is above, normal. This is a natural product. Let's just stick to what the product does, and let's make these outrageous statements. But then I started to see it in myself, and I started to see it in friends of mine. So we contracted with a lab in cosmetology, and essentially what they did is that they use very sophisticated photographic assays and colorimetric assays that measures color. And we measured the density of white in the hair of 6 individuals. We put them on Stem-Enhance for 3 months or 6 months, I think, and we quantified the density of white. And in every single individual, we saw a decrease. The average decrease was about 20% decrease in the density of white in the side and in the back of the head. And essentially, because we know that stem cells, first, when people were reporting these benefits, and I told them, please give me a break, I went on Medline, and I put stem cells and melanocyte. Can stem cells migrate in the skin and become melanocyte, which is the site making the pigments for the hair? And fair enough, documentation was there that stem cells can become melanocyte and can restore hair pigmentation. So that was the basis of the study, and we're using that claim simply because hair color is not a disease. And if stem cells can become anything in the body, I mean, hair color is just as far out as you can think. Nothing, as far as I know, is known that you can take orally that is going to reverse your hair color. So it is a very, very strong statement, because it's also a very strong symbol of rejuvenation. So it is, to me, a very, very strong statement to really what stem cells can do in the body. If it can reverse your hair color, it's an amazing statement of this rejuvenating capability of stem cells. But from there, you can extrapolate and say stem cells can become virtually anything in the body. And really, the idea is just to give it a try and see what it can do for you.
Okay, now because that was going to be my next question. I mean, you can't help but notice, looking at you, you've got quite a bit of gray. So it is working for your hair as well.
It is working for my hair as well. It's been, I travel. Actually, I must tell you, David, it's kind of a.
David, we want him to come back.
It's kind of an interesting phenomenon, because I travel I travel a lot. Probably one third to 1/2 of the year. It actually closer to 1/2 to 2/3 of the year I'm out of the country, and at times I just run out of product. And when I run out of product, at times I just, forget to take another bottle when I'm at the office for a day or two. And when I go without the product sometimes for 2, 3, 4 weeks, I go for the next haircut, and I can see that my hair is grayer. And then I go back on the product, and I take a lot of it, because I just have to go in the warehouse and take a few bottles. And when I get back on it, like with a vengeance, then I go to the next haircut, and I can see that the gray is going away. So that picture was taken on a day, I guess, that I've been traveling quite a bit, because I think that right now it's a little bit less.
Okay, all right. I apologize. Yes, we do want you to come back, because this is just fascinating for sharing. So I take that back. All right. Do you have? Is there a place where they have testimonials? I know nutritional companies kind of like have testimonials about what the products do. Is there a place that people can find this?
There are places where we're having some testimonials. I think if you go to teamstemtech.com, you will have some testimonials. But generally speaking, quite honestly, In a newsletter that we have some testimonials, but we really are extremely conservative. Probably one, if not the most conservative companies out there in terms of testimonials, simply because we're bringing forth here an entirely new concept. If you go on the web and you just plug stem cells, what will come out are things like the use of stem cells for reversal of muscular dystrophy and Parkinson and Alzheimer's. So the topic is so linked with diseases that we're trying to stay as conservative as possible to avoid any kind of misleading information. So we have tons of testimonials. Believe me, I could share some of them that are just unbelievable. But we don't go there. We let people just basically say how it changed their quality of life. So these testimonials are really benign. All I can say is that if people want to try it, it is one of the most life-changing product that I'm aware of, and I've studied a lot of them. I've worked with a lot of products. I know that I look certainly biased because it's my product, but I'm saying this from a very objective standpoint. Give it a try, and it has the potential here of really helping a lot of people.
Would you say that this could be the fountain of youth?
Well, if I make that statement and it's taken in any way by saying this product is a miracle, then I would be making some sort of an illegal statement. So let me stay out of answering the question directly, but let me just say that the moment we understand what stem cells really do naturally in the body, from that moment, I think that anything that is going to support stem cell physiology in the body is called to have a very significant impact on health. And that's why I believe that it's truly the future of health. In general, the book Cracking the Stem Cell Code, my aim with this book is to put out there this information in such a strong way and well-documented way that it's not going to become a discussion as to is it true, is it not true. It's there, 400 scientific references. It's clear the phenomenon that is being described in that book. Once we take that as granted, it opens an entirely new way of looking at health and wellness, because right now, if you look at any disease whatsoever, all we do is that we compensate for things. You lack insulin, we inject insulin. You lack thyroid gland, you give them Centroid. You lack dopamine in your brain, we give you L-Dopa. And I will go to the extreme. You know, you have spinal cord injury, we compensate with a wheelchair. But that's what we do. We compensate any problem with something. We rarely really get rid of the problem. It's not something that we do. And here, by discovering what stem cells do in the body, not only do we have a whole other way of tapping into the natural ability of the body to repair itself, but we can tap into a true, I would not say preventive medicine, because preventive means I prevent the big dark disease down the road. So it's still disease driven. It means that I do something to avoid that loss of health, but I'm still not doing anything to promote optimal health. All of this discovery on stem cells bring us to this place where for the first time in our history we have a physiological phenomenon that allows us to target not avoiding the big disease or avoiding something, but really just talking about maintaining health and tapping into optimal health. I think that this concept has the ability of literally changing the way that we look at health and wellness as a society. That's my goal.
And that just taps right into really what we're about, optimal health and not just managing and preventing disease. As a matter of fact, I rarely use the word prevention because it sounds more like a defensive posture. There's only so much you can prevent, but if you focus on building strength, like we talk about optimizing health, as you mentioned, that's will get to the goal. And you can see clearly that stem cell enhancement does appeal to that.
Well, you can never reach optimal health if you shoot for avoiding a disease. That is not in itself a health strategy. It can keep you into a better quality of life, no question about that, but it's not about reaching optimal health. And by optimal health, I mean what you meant and what you mentioned at the very beginning of the presentation, which is body, mind, and soul. It's a whole unit, and if you really want to be optimal in life, it's more than just not having a label that gives you a disease. We're talking about something that is far superior.
Well, that's what the WHO says. It says it's a complete social and mental and physical well-being, and not just the absence of disease. So
correct.
All right. Is this new science being introduced into the medical textbooks?
It is way too recent. That's the aim with that book. That book is written to be, could actually be used as a textbook. I'm going to Ecuador Sunday to lecture at the College of Medicine of Ecuador to the med students, mandatory lecture, because this concept is coming out right now. We need to understand that there is a bridge or a gap between research, academia, and then even a bigger gap between research and clinical application. We are right now in that transition phase. It is such new research that textbook like what I'm talking about here of the natural role of stem cells in the body do not exist. But textbooks in terms of what is the physiology of stem cells and how you can take them and proliferate them and their ability to become cells of various tissues, textbook of that nature, yes, do exist.
That's so sad. That's so sad that it takes a long time for medical students to learn that.
Well, I mean, I don't know if I would say. I mean, I understand what you say. You know, because this transition phase is creating a disadvantage for some people, but it's a natural progression of science. You need to. I mean, what we saw 9 years ago, 8 years ago, as being really what appeared to be where the field of stem cell research was going was really not that obvious. I mean, it was obvious in the sense that once you see a study where stem cells are shown to become brain cells and liver cells and it's happening naturally, to me, all it means is that we just need to further document it to a point where it becomes, you know, beyond the shadow of a doubt. And that's where we are now 10 years later. And it's at that point that then it starts to be taught, you know, in education, you know, institutions. So it's just a normal progression of science. It would be, I believe, it would be not unwise to start to teach things that suddenly it turned out to be not accurate and false. So it's just a normal progression of science. But we are at that place right now where that kind of information is being taught, is beginning to be taught in college.
Now, you mentioned that AFA helps to stimulate the polysaccharides that help to direct the migration of stem cells to wherever they are needed. Now, someone asked a question about glyconutrients. I know you're familiar with it, but my first question is what are the polysaccharides specifically, if you can share about that you use that.
Well, you know, a polysaccharide is very close to what you would say a protein. It's a chain of amino acids. And so a polysaccharide is a chain of monosaccharides. Now, these various monosaccharides, they're different ones, and they all enter into different proportions and different organizations in a polysaccharide. So what are they made of? They're made of essentially all the various monosaccharides that are known. So that's what you find in the polysaccharide that is coming from AFA. So that's what it's made of. The actual structure that was published, I believe it's in 2000 or 2001. This was published. We did some work in that area, but the actual configuration and polysaccharide that we have in AFA so far is the only one that was shown to have an effect on migration of stem cells. We have started to work with other polysaccharides coming from other plants. I'm not at freedom here to share any of that data, but at this point, only the polysaccharide from AFA was shown to have that effect.
Got it. Okay. And it's a special combination of the monosaccharides.
Correct. Yeah, polysaccharides, yeah. They all have different specific effects, although in a general way they all have some sort of immune modulating effect, or a lot of them have immune modulation effects. So they support some certain function of the immune system, but then they all have their different specificity depending on their sources. So AFA, the one from AFA, or from blue-green algae in general, because you see the same thing from spirulina, will support the migration of stem cells from the bone marrow. The one from AFA simply being more potent than the one in spirulina.
Speaking about spirulina, we actually had a webinar on that as well. So that's really, I did not know it helps in stem cell production. Great. Or migration.
Spirulina does not have an effect on stem cell production as far as I know, and from whatever it is published. We tested it and we didn't see it, but we do see it on stem cell migration.
Migration, got it. All right. We mentioned a couple of things about, someone is asking what is the relationship between stem cells and neurotransmitter? Does it help to, does the enhancement of stem cells help to replenish neurotransmitters, for instance? And I think I know where she's going to with this question, but what do you think?
Well, stem cells per se are not going to replenish neurotransmitters. But if we think, again, if we go back to the basics of it, stem cells are the building blocks that are going to go and repair various tissues. So the neuron that is making a neurotransmitter could very well have been a stem cell before. So if a stem cell moves into a position like in the brain or somewhere in the nervous system to become a neuron making a neurotransmitter, then yes, the effect of these stem cells could lead to increased secretion of the neurotransmitter through this very normal physiological process. Now, that's one thing. If we talk specifically about stem enhance, then keep in mind that it has this molecule of love in it, PEA, that supports the secretion of dopamine and norepinephrine in the brain. So this can help the production, or at least the physiology of dopamine transmission in the brain.
Now, speaking about the molecule of love, I thought oxytocin, where does that come in? Oh, that's the bonding hormone, isn't it? Isn't that like a molecule of love? Oh.
Yeah, oxytocin is more the molecule of love if you want it from the mother's standpoint, like motherhood. You give birth and you're in love with your baby, because it also plays a role in triggering contraction, and it's really around the birth phenomenon. It's not a hormone that is being secreted normally by the brain like in a normal life of a person.
Okay, well, because I was told that, I learned somewhere that if you hug someone, oxytocin is released by the lady for some reason.
Oh, well, I'm not going to say no. It's not my field of expertise, so I can tell. But from the basic, my background is neurophysiology. From the basic neurophysiology that I've done, 15 years ago, oxytocin is really a hormone more linked to or around the birth cycle. And if you really talk about the main neurotransmitters that are known to be involved into what you would call affective behavior, feeling good, love, and all of that, most of the information will be around dopamine, norepinephrine, and to some extent serotonin.
Okay, got it. All right, you mentioned something about immunomodulation. So something I had studied was that when there's an injury in any part of the body, there's a macrophage that migrates there and sends off the chemical signals that call upon stem cell production. In your talk, you mentioned that it was the damaged tissue itself that sends out the signal. How do we reconcile these two?
All of the above. There's nothing here to be reconciled. Macrophage will signal to their buddies from the immune system, but the effect on all the immune cells will come from both specific effect from macrophages and other local immune cells, but likewise from the tissue also. The tissue will also release compounds. A lot of these compounds will come from arachidonic acid that will basically be metabolized into this whole family of prostaglandins, and all of these are coming from the damaged cells. And it likewise also will come from the immune cells. SDF1 is produced by the injured tissue. It could also be produced by immune cells, but essentially most of the research at this point has been done by the tissue itself.
Got it. All right, someone's asked a very provocative question. Will this take the place of all embryonic stem cell use?
Well, it's a very, it is in itself a very simple question, although it can easily be made ambiguous. It can easily be a confusing question. Yes, it could, in the sense that, well, let me put it that way. A lot of what we know about adult stem cells was first done on embryonic stem cells before we got enough information to move on with adult stem cells. So I would be very dishonest to say we don't need embryonic stem cell research. Adult stem cells, alone is fine. There's a lot of information that comes from embryonic stem cell research. When we go from a clinical standpoint and the application of stem cells for health, then in that application, I believe that by all means, the future is adult stem cells, and the use of embryonic stem cells could be in very narrow application of gene modifications and things like this, for which even there, I am not sure that embryonic stem cells will be used or will be necessary. Now, it brings another aspect, which is the use of embryonic stem cells. Obviously, we want to stay away from it because there's a real controversy and ethical issue about it. And I just want to address one thing here. I do not want to take any side whatsoever. I think that one thing that has been a big disservice to our society as a general is the amount of misinformation that was carried in the media over the past 10 years or so, to a large extent, I believe, for political reasons and for a political agenda. Embryonic stem cell research was largely demonized as saying we're killing human babies, human embryos. It's very negative. These are human lives, and they have right to life, just like any human being, and all that kind of language. I'm not even going to express an opinion about it, but what I think is important to know, which has never been mentioned in the media, or at least not enough so that most people are not even aware of it, is that these human embryos are coming from in vitro fertilization. They are not made and fertilized in a lab. Well, yes, in a lab, but it's an in vitro fertilization lab, not in a stem cell research lab. And these embryos, the typical practice in a in vitro fertilization clinic is that you take a number of eggs from the woman, a number of sperm from the men, and you fertilize a large number of eggs, 15, 20 eggs, and then you make them proliferate in a test tube, and then you stop their proliferation at a stage where they are ready to be implanted in the womb. And so the mother comes, the couple comes, we inject a number of eggs, which will be anywhere between 2 or 3, hoping that one will take. And you remember the octomom, or the mom that had 8 of them, that's because they put 8 of these embryos and all of them picked up. So she ended up with 8 babies. It's a very, very unethical practice to do that many, but it's very common to put 1 to 3, hoping that one will come up, and then sometimes people end up with triplets. But they do that. So the family goes, do this procedure, and then you end up with 1 or 2 or 3 kids. You do that 1 or 2 or 3 times, and at the end, let's say after you've gone through 10 of these embryos, you have your family, you don't want to do it anymore. You're left with, let's say, 10 of them frozen in that freezer. What do you do with these embryos? Very, very simple. They're thrown in the trash. They're put into the sink and just trashed. Nothing else is done with them. People in stem cell research are coming and saying, instead of putting them in the trash, I will use them to do research and maybe help other people. So really what we're talking about here is human embryos going through death by disposal or death by research. That is really the question. If we say that we should not kill any human embryos, then really the culprit is in vitro fertilization. The question should be, should we create human life in a lab to start with? That is the real question that we should ask as a society. Once they're produced, they're going to die anyway. So I think personally that it's much more ethical to use them to serve another life rather than simply throw them in the trash. I think that it's a much greater moral position to take, but generally speaking, I think that where we are today in science, yes, adult stem cells is in a position to pretty much replace most of what our needs are for embryonic stem cell from a therapeutic standpoint. So it should put, or as we move forward, it should put an end to this whole dilemma.
I love the way you answered that. That was very good. Thank you. I'm sure that clarifies things for people. Somewhere in you probably answered this, but I have to ask if you're at liberty to answer, please go ahead. How much fold increase does this technology increase stem cell production?
Are you referring to stem enhance?
Yes.
Stem enhance would increase on average 25 to 30%, but we really have seen a range from just a few percent point to up to 1,600%, so 16-fold. So we have seen everything in between. But when we did analyze the data, we wanted to be extremely, extremely, extremely conservative for the work not to be attacked and criticized in any way. So we removed all these instances or studies or people in whom we saw these huge increases. So the true increase, if we really include all the data points, is closer to about a 50% increase. But if we remove all these eye data points, we have a conservative increase of about 20 to 30%.
And that 20 to 30%, that's just half of the story, because the other half, of course, is the stem cell migration.
That's correct. The only reason why we focus our message or the information that we have mostly about the release is that there is very, very little documentation of the effect of migration. What I mean here is that if you remember the study that we shared about Orlick at the NIH, where they triggered heart attack and they simply increased the number of stem cells in the bloodstream, they migrated to the heart without any support. The only support was increasing the number of stem cells. If you do nothing, you don't get that benefit, but they still have the ability to migrate. So migration does not appear to be as much as a key phenomenon in the whole process. I believe that it does. I believe that it is. Once you support the release, I do believe that it is playing a very relevant role and significant role in the whole picture. But it's very poorly documented at this point. So we keep the core message on the release of stem cells. Very little has been done, and there's very little we can say right now about the real significance, physiological significance of the migration part.
Got it. Got it. Well, this has been fantastic, folks. Again, thank you, Christian. And I'll actually put in a message here saying, thanks, Christian, for sharing the redeeming side of the embryonic stem cell research, which we rarely hear from the media. As a result, our blind spot grows. Thanks again.
Enormous amount of misinformation. You know, when we think about anything that is said, like, you know, pictures were even circulated where they were showing that embryonic stem cells meant the killing of fetuses and babies. And I mean, we're talking here about a lump of cells that has about 8 to 100 cells that is so small that it's invisible to the naked eye, and it's the same thing that that many women in their normal life are simply passing when they have their period. So it's totally invisible to the human eye. So when we're going down to the side, to the point of saying we're going to call destroying that human embryo a crime, that means that, for example, alcohol drinking that was shown to reduce the implantation of a natural implantation of a human embryo in the woman's womb, that would mean that drinking alcohol could make a woman criminal. I mean, this concept here of that was brought in the media had such far-reaching implication that honestly, it was mind-boggling to see that level of misinformation. And hopefully we will have one day a forum to address that in the media, not to defend embryonic stem cells. I couldn't care less about defending embryonic stem cells. I just think that we need to bring sound information. People are smart enough and strong enough to handle what's the right information, and to tweak that for political agenda, I don't know, it just rubs me in the wrong way.
Right. Well, okay. Looks like all the questions have been answered except for the disease conditions. So we're going to leave that out. But again, thank you, Christian. We definitely would love to have you to come back. I know we've mentioned a couple of other topics. We definitely need a follow-up on this webinar. And we want to talk about the other topics. Accelerated learning. I know you mentioned accelerated learning. We've had someone speak about accelerated learning for kids.
Okay.
We need someone to come speak about accelerated learning for adults as well. So hopefully come and cover that up for us sometime.
Very good.
Well, again, we appreciate this. Thanks, folks, for joining us on this webinar. If you have any questions, go to our website, buildingstrengthwebinars.com, for a follow-up. And if you want to try the product, again, the website or the information is right here. You can call and contact Heather. The webinar is coming up next week, Navigating Uncertainty, and Dr. Coolish will be on to speak about irritable bowel syndrome and Crohn's disease on Monday. This has been a great webinar. We're going to wrap it up here and say good night, and thank you for joining us all. God bless. Bye.
Good night.